Have you ever wondered how our body repairs its DNA? It's a question that may not cross your mind daily, but the answer is a fundamental part of our existence. You see, our DNA is not invincible. It suffers damage from various sources, such as environmental exposure and natural radiation, and even during the process of cell division. But fear not, our body has an ingenious system for fixing this damage and ensuring the stability of our genetic information. At the heart of this system is a protein called RAD51, which plays a crucial role in the DNA repair process. It works alongside a biological process known as homologous recombination, an intricate dance of genetic information exchange that helps to mend the broken strands of our DNA. It's a fascinating mechanism, to say the least. So, what is this RAD51 and how does it function in our body? Stay tuned as we delve into the mysterious world of DNA repair. RAD51 is a gene that encodes a protein that plays a crucial role in DNA repair. This gene is like a soldier on the front lines, continuously working to maintain the integrity of our genetic information. Imagine RAD51 as a detective. Its primary function is to find and invade homologous DNA sequences. Why does it do this? To enable accurate and timely DNA repair. When DNA is damaged, it's like a puzzle with missing pieces. RAD51 helps by finding matching sequences in the DNA, serving as a template to fill in the missing pieces and complete the puzzle. Now, RAD51 doesn't just find the matching sequences, it also binds to the DNA at the site of a break, encasing it in a protein sheath. Picture it as a first responder at an accident scene, putting up a protective barrier around the damaged area. This encasing is an essential first step in the repair process, like securing the scene before the actual repair work begins. Breaks in DNA can occur due to various reasons. They could be caused by natural and medical radiation, environmental exposures, or even during the exchange of genetic material in preparation for cell division. In all these scenarios, RAD51 steps in to initiate the repair. But RAD51 doesn't work alone. In the nucleus of many types of normal cells, RAD51 interacts with many other proteins, including BRCA1 and BRCA2. Think of these as a team of experts called in to fix the damage. The BRCA2 protein, for instance, regulates the activity of RAD51 by transporting it to sites of DNA damage in the nucleus. The relationship between the BRCA1 protein and RAD51 is a bit more complex. Although research suggests that BRCA1 may also activate RAD51 in response to DNA damage, the exact mechanics of their interaction are still being unraveled. By helping repair DNA, these three proteins, RAD51, BRCA1, and BRCA2, play a vital role in maintaining the stability of a cell's genetic information. They are the unsung heroes in our cells, tirelessly working to keep our genetic blueprint intact. Now that we know about RAD51, let's delve into homologous recombination. Homologous recombination is a type of genetic recombination that allows for the exchange of genetic information. This process is an essential tool in the toolbox of our cells, enabling them to accurately repair the harmful DNA breaks that can occur on both strands of DNA. This type of break is known as a double-strand break, or DSB for short. Imagine a highway with traffic flowing smoothly in both directions. Now, picture a massive pothole appearing, disrupting the flow in both lanes. This is akin to a double-strand break in our DNA. Homologous recombination is like a highly skilled road repair crew that comes in to fix the damage and restore smooth traffic flow. But the role of homologous recombination doesn't stop at repair. During meiosis, the process by which eukaryotes make gamete cells, like sperm and egg cells in animals, homologous recombination shuffles the deck, producing new combinations of DNA sequences. This reshuffling is a key contributor to the genetic variation we see in offspring. It's like nature's way of ensuring that every new generation has its own unique genetic blueprint, enabling populations to adapt during the course of evolution. Now let's take it a step further. Homologous recombination is also used in horizontal gene transfer, a process that allows different strains and species of bacteria and viruses to exchange genetic material. This process is the primary mechanism for the spread of antibiotic resistance in bacteria, essentially allowing them to share survival strategies and adapt to threats. So, you see, homologous recombination is not just a repair mechanism. It's a versatile process that underpins life as we know it. From the stability of our own cells to the evolution of species. And at the heart of this process is RAD51, the protein we discussed earlier, which finds and invades homologous DNA sequences to enable this accurate and timely DNA repair. So homologous recombination and RAD51 work together in maintaining our DNA stability. RAD51 and homologous recombination form a powerful duo in DNA repair. Together, these two biological marvels work in harmony to maintain the stability of our genetic information, a feat of paramount importance to our overall health. Let's take a quick recap. RAD51 is a gene encoding a protein that plays a crucial role in homologous recombination and DNA repair. This protein finds and invades homologous DNA sequences, enabling accurate DNA repair. It binds to the DNA at the site of a break, encasing it in a protein sheath, which is an essential first step in the repair process. DNA breaks can occur due to various reasons such as natural and medical radiation, environmental exposures, or during the exchange of genetic material in preparation for cell division. On the other hand, homologous recombination is a type of genetic recombination where genetic information is exchanged between two similar or identical molecules of DNA. It is widely used by cells to accurately repair harmful DNA breaks that occur on both strands of DNA, known as double-strand breaks, in a process called homologous recombinational repair. This process also produces new combinations of DNA sequences during meiosis, which generates genetic variation in offspring. In the nucleus of many types of normal cells, RAD51 interacts with other proteins, including BRCA1 and BRCA2, to fix damaged DNA. The BRCA2 protein regulates RAD51's activity by transporting it to sites of DNA damage in the nucleus. The interaction between the BRCA1 protein and RAD51 is less clear, but research suggests that BRCA1 may also activate RAD51 in response to DNA damage. By working together, RAD51 and homologous recombination not only repair damaged DNA, but also contribute to maintaining the stability of a cell's genetic information. This is critical because any disruption in this stability can lead to genetic disorders and diseases. So, next time you think about your body's amazing capabilities, remember RAD51 and homologous recombination, the unsung heroes of DNA repair.